Hello everyone, welcome to Nalanda Advantage. Today we'll learn about different topics. Some of them are endoplasmic reticulum, the structure and function of ER, ribosomes, Golgi bodies, lysosomes, and important questions related to the topic. So let us quickly start. This ER or endoplasmic reticulum is a network of hollow membrane tubules. As you can see here, these tubes, they connect to the nuclear envelope and the cell membrane. The major function of endoplasmic reticulum is to function in the synthesis, modification, folding of the cell products and thereby transporting it to their respective locations. There are two types of ER, which is known as rough ER and smooth ER. Now let's see about rough endoplasmic reticulum. Since the name rough arises because of the ribosomes attached on the surface of these endoplasmic reticulum, hence it gives us a rough appearance. The proteins synthesized by the RER have specific final destinations such as the cell membrane or the exterior of the cell or even the ER itself. So basically the ribosomes attached on the surface layers give it the rough appearance. Now let's learn about smooth endoplasmic reticulum. The term smooth means it is devoid of any ribosomes attached to it. Basically, it's a smooth layer without any ribosomes attached to it. SER is involved in the synthesis of lipids, which includes cholesterol and phospholipids, which are used in the production of a new cellular membrane. It also regulates calcium in the muscle cells as well as it destroys toxic substances with the liver. Now let's know about more about ribosomes. These ribosomes, they are made up of proteins and RNA. They are the functional unit. Basically, they are required for protein synthesis. They are also known as the protein factories of the cell. The ribosomes, the smallest unit of a proteins, they are made up of amino acids. So they join these amino acids to make proteins, the process known as protein synthesis. It's a very unique machinery which involves the use of transcription and translation. Ribosomes, they are discovered by George Ballard, and each ribosome, it is basically composed of two subunits, a larger one and a smaller one. Each one has a characteristic shape also, and these subunits, they are different in eukaryotes as well as prokaryotes. The small and the large subunit of eukaryotes are designated as 40S and 60S respectively. This S or the capital S that you're seeing here, this S is basically the Swedberg unit, which is the coefficient that we get by centrifuging these subunits. While prokaryotes contain small 30S subunit and a large 50S subunit. So this is the major difference between the eukaryotes and the prokaryotic subunits. As you can see here in the diagram here, given in the diagram here, the amino group, this is a small representative of an amino acid. These amino acids, they get converted into your linear chains or your protein machinery, and basically ribosomes are responsible for that. Now let's learn about Golgi bodies. As you can see in this diagram here, they are the stack. They are stacked one over each other, and they have a shipping site, a transphase as well as the receiving side, which is known as the cis phase. They receive proteins which are made by the ER, and they transport vesicles which are modified proteins, and they pinch off at the ends. So basically, these are the few transport vesicles. They are associated with the ER, and all the vesicles or any of the molecules that get transported is done by Golgi bodies. So basically your ER and Golgi bodies function as your delivery mechanism or the delivery system. Like we had for whenever we do online shopping, we get any products delivered to us. It involves lot of the stoppage. Similarly, your ER and Golgi bodies also, they act as your delivery mechanisms. Now let's see a good animation where materials are transported from your rough endoplasmic reticulum to Golgi to the cell membrane by vesicles. Over here we can see the transport, synthesis and modification of this entire vesicles out of the cell membrane. So this is how your Golgi apparatus works, from ER to the Golgi apparatus and delivered to the respective location where it is required. Next organelle is lysosomes. Lysosomes, they contain digestive enzymes. They are also known as suicidal bags because they break down food, bacteria and worn out cell parts for the cells. So anything which is toxic and is not required, basically they get metabolized in this. They are programmed for cell death or autolysis. They lyse or break open and they release enzymes to break down and recycle the cell parts. Now let's see how this digestion works. As you can see in this figure here, this is a figure of a cell. They take the food, a food that is present by the phagocytosis. Phagocytosis is a process in which the food is get engulfed wholly by the cell. It digests the food and they get rid of the waste. This is the quiz time, so let's take the first question. Which of the following is the largest single membrane bound intracellular compartment? Is it ribosome, Golgi apparatus, nucleus or endoplasmic reticulum? So the answer is D. Endoplasmic reticulum is a network of closed and flattened single membrane bound intracellular structures, and this ER lumen is a closed compartment between ER membranes which are physiologically active. Next question. Protein undergoes modification in ER before reaching their final destination. Is this statement true or false? The answer is definitely true. Now, after the synthesis of a protein, it undergoes various modifications before reaching their final destination. And the main process is done by ER and Golgi. Now, let's see the third question. What is microsome? Is it a compartment of Golgi, a smaller ribosome, or a small ER compartment, or a small vesicle of a fragmented endoplasmic reticulum? The answer is D. Since microsomes, they are the small vesicles formed by the breakdown of ER membrane into fragments. They are derived from RER, also known as rough microsomes, while few of the microsomes lacking ribosomes, they are known as smooth microsome. So I hope the topic is clear to you. It was a small video consisting of ER, lysosome, ribosomes and Golgi. They are close-knit associated functioning organelles in the cell. Thanks for watching. Please subscribe the channel and share, like and comment. Your valuable feedbacks help us to grow. Thank you so much.